To understand the process of translation, that is protein synthesis, first structure of tRNA and function of it or role of tRNA in translation must be understood, as well as role of other RNAs. We know what is the full form of tRNA? That is transfer ribonucleic acid. So full form of tRNA, transfer ribonucleic acid. Now coming to parts of tRNA involved in translation or protein synthesis. First one is anticodon arm or anticodon loop, and second one amino acyl arm or loop, amino acyl arm or loop. Now, how they are involved in translation? First, let me show. This is amino acid attachment arm. This one is amino acid attachment arm, and this is the specific amino acid attachment site, and this one is anticodon loop or arm. So it's better to say this one is amino acid attachment arm or loop. Let me write it. Amino acid attachment arm or loop. Now what is the function? From the name only we can understand. So anticodon arm or loop, it has specific anticodon, specific or complementary. Anticodon arm have complementary bases to the codons of mRNA. So anticodon arm of tRNA contains the bases which are complementary to the bases of mRNA, that is called codons. Okay, so tRNA can bind with mRNA due to complementarity. Now coming to next part, that is amino acid attachment arm or loop. Actually this must be discussed prior to anticodon arm or loop. As we can see, at the 3' OH end, one amino acid, that is 3 bases or 1 nucleotide, comes and joins. I repeat, at anticodon arm, at amino acid attachment site, that is 3' OH end, 3' OH end of tRNA. At the amino acid attachment site, one codon or nucleotide, we can say ribonucleotide, comes and joins. Later, during translation, it is added to the growing amino acid or polypeptide chain. I repeat, at amino acid attachment site, that is 3' OH end, one codon or nucleotide, that is 3 bases, come and join. According to the, and later this codon or this amino acid, we can say one codon codes for one amino acid, that we studied in genetic code. This, what it does, this amino acid attaches itself with the growing polypeptide chain during protein synthesis. Now next part, that is, what is initiator tRNA? It is a specific or special type of transfer RNA which binds with specifically codon AUG for specifically codon AUG which codes for methionine. Then after binding it, then this tRNA starts the process of protein synthesis. Okay, so till now we studied parts of tRNA involved in translation, function of each part. What is tRNA? Initiator tRNA. Now what is secondary structure of tRNA? That's the next point. Here you can see this is the primary structure. This one, linear, as you can see, it's single stranded linear structure. Then it forms, when it folds, it forms this kind of a structure. This structure. This is secondary structure of tRNA. So the folded structure of tRNA due to formation of H or hydrogen. H means hydrogen bonds at specific location is called secondary structure of tRNA. Now coming to how does it look like? It looks like clover leaf. One more point, who discovered tRNA or who proposed this model? That is Frederick Crick. Francis Crick. I repeat, who proposed structure of tRNA? It's Francis Crick. I repeat, Francis Crick proposed structure of tRNA or cloverleaf model of tRNA. Now coming to function of tRNA. So what is the function? tRNA acts as adapter molecule. Now what it is? Adapter molecule means that is, or i.e., that it with tRNA amino acids in form of codon, amino acids come and attach at amino acid attachment site, that is 3' OH end of tRNA. Later this amino acid joins with growing polypeptide chain during translation, that is protein synthesis. So that's the main function. So one by one tRNA goes, attaches with the mRNA during translation, and one by one amino acids are added to the growing polypeptide chain. That's the main function of tRNA. In simple words, tRNA acts as carrier of amino acids during protein synthesis or translation. When we discuss about the steps of translation, this can be understood better. Now, how tRNA actually looks like? It looks like inverted L. Now coming to translation. Definition of translation. What is translation? So let's discuss. Translation is the second step in central dogma. It is continuation after transcription, as we can say, and during this step amino acids are added one by one to form polypeptide chain. Another term for translation is protein synthesis. So is the second part in central dogma in which polypeptide chain is synthesized in rRNA in a ribosome using mRNA as template and tRNA as carrier of amino acids. So from this only we can say role of mRNA and tRNA. So first one, role of mRNA. mRNA, what it is? As we can see, acts as template. What does it mean? According to the bases present in mRNA, tRNA carrying a specific complementary amino acid or anticodon binds with mRNA. So in simple words, the bases present in mRNA, according to that, a specific type of tRNA carrying complementary bases to the basis of mRNA comes and binds by forming hydrogen bond. Now, role of rRNA. Steps will be discussed in the next discussion. So rRNA, what is the role of rRNA or ribosomal RNA or ribosome? ribosome, specifically the small subunit, acts as site for translation. Actually, ribosome consists of two subunits, large and small. During protein synthesis or translation, among these two, in inactive form, ribosome has two subunits, large and small. When mRNA comes and binds with the small subunit, it gets activated and then protein synthesis starts. So these are very important. Both mRNA, tRNA as well as ribosome are very essential for the process of translation.